problem that has crippled the whole globe. How to find form at the topic for today's webinar and its implications in the present pandemic holds lot of promise in terms of research, which eventually will help in addressing treatments because nothing can happen without understanding what is really the problem. Only then the solution can be thought of. We have been having rainfall in Bangalore, but what has caused all the disturbance, both in the district of Dakshina Kannada, Malnad, as well as Uttar Kannada, the Udupi districts, have been the storm. Rain in quantities that can prove to be a boon have become a bane, have become a serious problem because of the storm. And the storm has resulted in excessive rainfall, crippled life, resulting in many deaths. So to understand cytokine storm, it's very easy for us to compare what is now happening with what is happening in the pandemic. Cytokines have always been considered molecules that are very important in warding off many kinds of viral conditions, but when it became a storm, then it resulted in all of the pathologies that got associated with it. Way back in 1976, when I first began my research working on cytokines, I really didn't have too many people at that time talking about it because I was looking at the role of cytokines in parainfluenza 3 virus infections. And that time when we studied the tumor necrosis factor, the macrophage migration inhibition, macrophage aggregation, leukocyte migration inhibitions, little did I know that the same molecules that were being understood in a very primitive fashion then, accounting for only the way they managed a viral infection, would ultimately be researched upon to an extent that they have caused all the havoc and the disabilities that are being witnessed in the pandemic is mainly due to the storm. So be it our thunderstorm, rainstorm, the sandstorms, a storm is always something that results in devastation. Okay, that is a little bit about the topic for all those who are online listening to what are cytokines and what are going to be cytokine storm issues, which will be, of course, dealt with by the expert. I will proceed to introduce today our guest, our opening speaker, who will introduce our university also, Professor Harsha Halahalli. Who best can you have for an event than someone who is an educationalist, who is a researcher, and most importantly, a gentleman par excellence with humility unparalleled. That is the word I would like to use for this gentleman, Professor Harsha Halahalli, whom all of us know as someone very approachable, very knowledgeable, and very, very objective in everything that he does. I would like to inform the audience, many may know him, but realizing that we are going to have audience from all over the world who have registered, even if they are unable to be online to listen at this very moment due to differences in the timings, they might all be able to later listen to this through the Nitte YouTube channel. And therefore, it is not out of place for me to describe a little about our own Professor Harsha Halahalli. Having got his medical degree from the Karnataka Institute of Medical Sciences, KIMS, as we popularly called in Hubli, he proceeded to do his master's in the subject of physiology. For a researcher, the subject of physiology is really the icing on the cake. Any medical researcher, and all of us know the Nobel Prize is usually given in medicine and physiology, and he went on to do that in the prestigious institute at Pondicherry, the Jawaharlal Institute of Postgraduate Medical Education and Research. And there was no looking back, having done his MPhil and his PhD from JIPMER. He moved on, MD from JIPMER. He moved on to taking up a job at Manipal, the Kasturba Medical College, but it was not going to satisfy him because research has been his passion. He went to Bangalore to the National Institute of Mental Health and Neurosciences, NIMHANS as is popularly referred to do his PhD. And of course, before that, he did his MPhil too. So the levels at which he acquired all his knowledge speaks volumes for his own passion in research. And that passion sees a lot of contribution in neurosciences and neurobiology of psychiatric disorders. Particularly, he correlates, of course, a lot of the neuroimaging and electrophysiological work in schizophrenia and neurobiology of high altitude induced cognitive deficits. Well, these words are exciting for people who would really know what a volume of research remains to be done in such a lot that is already explained, but large volumes that remains unexplained. Well, this is his past, but that has been the good fortune of Nitte University that we have him with us as the curriculum director at the moment, but I know he would prefer to be referred to as the professor of physiology because of his own passion for teaching and research. And that enables him to do, of course, two things at one shot. One is his passion of teaching, which find appropriate application through the various digital platforms that he has created in this university as the curriculum director. Now, being an educator, adapting yourself to the use of newer technologies in medical education, adapting yourself to developing them, not just using, but developing newer platforms, both for teaching, for examinations, for research deliverance, not doing research sometimes yourself, but understanding the problems of researchers and enabling through the position that is given to him to lead teaching and research and medical education through blended learning. We at Nitte are most fortunate to have this young, dynamic person in Dr. Harsha. For me, he's Harsha, but for everybody else, I'm sure Professor Harsha Halahalli is an embodiment of everything that is perfect. So I think if you let me talk more about him, I can talk for ages, but I will at this moment hand over to describing our guest. And I now request, of course, Professor Harsha, if I think he's ready there to give his opening remarks. Harsha, please. Dr. Harsha? Yeah.
Professor Indrani Karunasagar, thank you for those extremely kind words. Dr. Indrani has always been very effusive in her praise and introductions for me. And I'm afraid that probably my introduction to this webinar is going to be shorter than her introduction to me. So, let me begin by welcoming Dr. Ram Savan from the University of Washington, Seattle. We are delighted to have Dr. Ram with us here today. I welcome him on behalf of the university and on behalf of NUCSER, the organizing institution here. I've heard of Dr. Ram from Dr. Indrani and Dr. Anirban earlier, even before this webinar, and I'm sure that he will bring great value to this webinar today. I would also like to welcome all the delegates who have joined us from various places today. Welcome to this webinar series of Nitte University, and we hope that you have enjoyable session today. The Nitte University Center for Science and Education Research, which is organizing this webinar on behalf of Nitte Deemed University, is a young institution, but a very dynamic and productive institution. A small band of scientist teachers in this institution, many of them who are quite young, led by very experienced and senior researchers like the two Dr. Karunasagars that we have, Dr. Idya Karunasagar and Dr. Indrani Karunasagar, have done wonders for the university. As a university, in a matter of a dozen years, we have been consistently ranked in the top 70, 80 within India, and we have also, figuring in the top 500 in Asia. That's no mean achievement, without sounding modest. But I would like to point out that NUX's contributions to this has been immense. Particularly in the field of research and in promoting innovative science programs. Our NUXer, we consider it as a jewel in the university's crown, and we congratulate them in organizing this very topical webinar. Coming to today's talk, I'm sure that what Dr. Ram is going to talk about today will resonate with everyone here today. Interferons and cytokines are at the heart of immune responses, especially in viral infections. Over the decades, they have been recognized as the key players in orchestrating a very complex response of both antibody-mediated and cell-mediated immune responses in various conditions, especially as the defenses against viral infections. The number of cytokines that are being recognized and characterized is growing exponentially. When I started studying medicine 25 years ago, there were a handful of cytokines, and the term cytokine itself was a very novel term. But I have seen over the years that hundreds and thousands, if not thousands, of cytokines have been identified and characterized. This growth of the cytokine biology has been so immense and overwhelming in some senses that traditional approaches, descriptive approaches to this part of physiology and immunology are sometimes becoming inefficient. And there is even a move towards mathematical modeling to understand the network of cytokines, the interactions of cytokines which drive many of the immune responses in various conditions. Obviously, when such complex interactive systems are there, they are likely to go awry, they are likely to become dysregulated and disordered. And possibly in case of COVID-19, such an inappropriate and possibly excessive activation of cytokine responses has been recognized as an important element in the pathophysiology of the disease, particularly in cases where the disease progresses rapidly, where there are multiple organ failures involved, and of course, where fatalities are recorded. And the term cytokine storm has now become, a part of our everyday lingo. It's become popular even in the lay press. And Dr. Indrani gave a very, very apt and a very intriguing analogy about, the monsoon storms that we have and the cytokine storms, both of which are essentially, normal, which are essentially required, which are essentially useful, but when they cross the threshold of a particular threshold and gain momentum, they become damaging. So this is a very relevant topic, and I'm sure that Dr. Ram's topic will be of interest to everyone who has gathered online today. Nitte Deemed to be University, of course, has been in the thick of action in this pandemic situation. Being a health science major university, we have been in the center of the COVID storm, so to say. The clinicians, of course, got into the act early. There has been a major reorganization of hospital services. We are proud that our clinicians put together very detailed and very well thought out protocols very early on when the outbreak hit our country, and even before the cases started rising in our part of the world, in Bangalore, we were prepared for it. And we have also been very good at adapting to the rapidly changing scenarios. Our teaching hospital was one of the first hospitals in this region to get approved RT-PCR lab, and it handles a large chunk of the diagnostic work that is required for our district and neighboring districts. And Nuxer played a role in that. We have also gone on to set up special treatment centers, COVID care centers, and more recently a plasma therapy unit, one of the few approved plasma therapy units in this part of our state. We are home to it, and quite a few patients have benefited from such innovative therapeutic initiatives. And the researchers at Nitte University have also not lagged behind. They too have been very proactive. I understand that there are funded projects underway in the university in various institutions, especially in Nuxer, which are developing or refining rapid diagnostic tests based on variants of PCR. There are studies which are looking at the role of AC receptors in the pathophysiology of COVID-19. Looking at of course, the role of inflammatory markers and the diagnostic and prognostic applications, and of course, a large number of clinical studies, and also being home to a large number of students, we are also exploring the impact of COVID or pandemic situations in general on how education of students is impacted, how the mental health of students is impacted. So what I'm trying to say is that there has been a variety of various fronts along which research has been initiated and is going on in Nitte University. And therefore, webinars like this, which focus on this topic, are extremely useful for us internally, and we are glad to share this with external delegates to help in spreading the word and to help in educating extending education beyond the confines of our university. So let me wish everyone a very happy and good learning session today. Dr. Ram, welcome to this webinar. Thank you for having me. Organizers, I'm particularly grateful for giving me this opportunity to be part of this today. Thank you.
Thank you. So thank you very much, Dr. Harsha, for your opening remarks. So you have very nicely highlighted the achievements of the university and the work that is being done here, both on the clinical side as well as research. And also as a physiologist, you have laid a nice foundation for learning about cytokines in health and in disease. So thank you very much for your excellent opening remarks. So now it's a privilege for me to introduce our brilliant speaker, Dr. Ram Sawan, whom I have known since his undergraduate days. So he did his bachelor's degree in the University of Agricultural Sciences, College of Fisheries, Bangalore. So then I came to know him. And then I knew him even more intimately when he joined the Department of Microbiology as a postgraduate student, and then he did his thesis with us. So it has been a nice experience for us to work with him at that time. And he showed his brilliance at that time, and his thesis work was adjudged the best thesis at national level. And he was awarded the V.G. Jingran Best Thesis Award for his master's work. And of course, he had set his goals very high and he wanted to go global. So he was looking for opportunities outside, and he was awarded the Mumbashu Fellowship at the Miyazaki University for his doctoral studies. So he went to Miyazaki, where he started his studies on immunology. Of course, cytokines are part of the innate immune system. So the best way to study innate immunity is to start with the invertebrates and then lower vertebrates and have this comparative immunology background, which will help one to understand how the organisms have been adapting to the microbes around them, some of them which cause disease. So Dr. Ram Savan did his PhD there in Miyazaki University, and then he moved to the United States, to the National Institute of Health and National Cancer Institute on a postdoctoral fellowship. So there he spent more than 6 years, initially as a postdoctoral fellow, and then he got a tenure position at the University of Washington, Seattle. So even in NIH, he carried out excellent work, which was recognized by the institute, National Institute of Health, where he got two awards: Director Innovation Award of the National Cancer Institute of National Institute of Health in 2009 and 2010. So he showed his brilliance there, which led to his being recruited as a faculty at the University of Washington, Seattle. Initially as assistant professor, and then he rose to become the associate professor. And he, during his career, again he got number of other awards: Milstein Young Investigator Award, and he has been with the International Society for Interferon and Cytokine Research, and he has chaired a number of sessions uh, in international conferences. And presently, he is the co-chair and of the organizing committee of Cytokine 2020. So he'll be organizing this conference. So during his research career, he has reported many new interleukins, and if you look at his list of publications, it can be the envy of anybody, because he has more than dozen papers in Nature, and then also in other top journals like Cell and number of immunology journals. So no doubt he has been recognized in the United States and given the position of a leader uh, in the University of Washington, Seattle. And we are indeed very happy that he agreed to give this talk today. And now I hand over to him for this talk.
Okay, thank you, Dr. Harsha, thank you, Indrani ma'am and Kanasagar sir, because both of you have been my teachers for a very long time, and whatever I know today, I still tell my students that it's because of Mangalore Fisheries College and the microbiology department. So let me figure out how to share. And I know I got a primer for this. Can you see my screen?
Yeah, it's clear now. Yeah, it's moving on. Yeah, yes.
Okay, cool. Thank you very much. I really appreciate this opportunity, and I have plenty of friends at NUSER, and I once in a while look up NUSER to see who's there and how they're doing, and it looks like a lot of amazing work has been done at NUSER. So let me talk about one family of cytokines called interferons. Like Dr. Harsha said, there are so many cytokines, and we'll be sitting here for a couple of days to go through most of these cytokines. So what I've decided is to just talk about interferons, because this is very near and dear to me, and my lab works on interferons and viral diseases. So I thought I would give you a primer on how interferons work, what are the recent advances on interferons, and how are they controlling viral diseases, and the implications in COVID. Most of my work will be on different viruses, and you can see how these could be important for COVID-19 as well. So before I talk about the research, none of this work is done by me per se. I don't go into the lab and pipette. So it is apt to acknowledge all the wonderful people I have in the lab and the collaborators who have actually done this work. And it's a pleasure to run a lab here with really smart people all around the world. And most of the work which I'll be showing today is from Hannes, an acting instructor, Adriana Ferrero, who was a senior fellow in my lab, who has her own lab, and the rest of the work I'll be showing are not in my lab at the moment, who have all left it for other jobs. Okay, and also the collaborators and the funding. Okay, so immune system is in a never-ending race with pathogens. And why I say this is throughout the evolutions, right from common vertebrate ancestors from lamprey to bony fish, to mammals and the current human beings, what is common among all this is all these animals were infected with pathogens, right? It could be bacteria, virus, fungi, or other parasites. To ward off these pathogens, these organisms started developing a response against this. And if you go in invertebrates, you have the RNAi response, which was the primary form of defense against viruses. But also these pathogens, the host also developed a strong innate immune system. And this dates back to 600 million years, right? So initially you had a strong innate immune system. For example, macrophages, which were really important to kill bacteria. And then at the same time, you started seeing a much more sophisticated immune system called adaptive immune system, where the B cells and the antibodies arrived. And this is somewhere during cartilaginous fishes and bony fishes. And we talk about vaccines to COVID-19 and other diseases. And vaccines are primarily based on the adaptive immune responses. So during this time, when you had bony fish, there was a massive explosion of these innate immune genes called cytokines. There are plenty of cytokines, which we and others have found in bony fishes, but also this really intriguing molecule called interferons. So my lab has been interested in trying to understand how interferons and cytokines are important in warding off viral diseases. So we take a very different approach in understanding the importance of these molecules. And as you will see throughout the presentation, some of the secrets, what we think make us all different, how we combat viral infections, is hidden in our genome. And as you can see this picture where this is a painting of a girl with the smallpox, that means to say for as long as we have known human mankind, or we have seen bacteria, viral, parasitic diseases. But these have not defeated us because we still live as a species. That means to say, we have been constantly adapting, our immune system has been constantly adapting to these pathogens. So one of the bigger thoughts we have in our lab is, we all see viruses differently. A virus which infects me might be, my body might not able to fight it, but my friend who might have the same viral infection might just shrug it off. It might be as simple as a cold for him, and he will shrug it off and walk away. So the question is, what does determine these differences in the way we ward off these viruses? So if you compare an animal X versus animal Y, and you compare their DNA sequences, And if you align them, if you think all the 3 billion bases of our genome, if you compare them, what you see is single nucleotide changes. And this occurs all over in the genome. And if you compare our genome to my favorite species, which is the fish, 70% of our DNA are similar. Like if you align this, 70% of it is very similar, but 30% is different, right? But now if you go a little higher and if you compare humans and chimpanzees, 99% of our DNA are very similar. That 1% of DNA changes is the difference between us and chimpanzees, okay? And if you go further and compare humans to humans, 99.9% we are similar. That 0.1% determines our skin color, our differences in the smell, our differences in the taste, our differences in the hair, right? So my lab is interested in that 0.1%. And if this 0.1%, if this is important for the differences we see during viral infections. Okay, so to give you a much more simpler explanation, if you just think about an innate immune gene, what you see here is an mRNA, and this mRNA has a protein coding region and a untranslated region, which is the 3' UTR and the 5' UTR. And one can imagine these nucleotide changes, what I mentioned, the C to T nucleotide change, if it happens in a protein coding region, and you know, because of the triplet codon, you will see a change in amino acid, right? So an amino acid could change based on where this SNP is, this one nucleotide changes. And sometimes this one nucleotide change could be beneficial, right? This change could have changed the protein and it could make this protein work harder and better, right? But in case if this change will abruptly end the protein, right? Like a stop codon. And if it stops the protein, this protein might be non-functional, or you might generate a human knockout, right, of that protein. And that could have a lethal effect. Or a very benign change in amino acid could also cause this protein to be lethal, right? So this one nucleotide change could have multiple effects in the coding region. But what is intriguing is most of our changes in the genome is in the non-coding regions, and that makes sense because you don't want to take that chance where a change could be either beneficial or lethal or no change. That kind of dice, I don't think the evolution would play because there is a 1/3 chance that it could be lethal, right? So if you look at the genome, you see most of the changes in the nucleotide are located in the non-coding fraction of the genome, okay? And if you put all the non-coding fraction together, and the changes in the non-coding fraction of the immune genes is just 0.1% of our genome. And that is what my lab is interested in, okay? And what I want to show you with one example is how that 0.1% change or just one nucleotide change among humans could have an enormous impact in how we see a virus. Okay, before getting into that, I just want to give you a primer of what interferons are. So interferons are the first and the critical defenders to clear infection. As the name suggests, interferons meaning interfere, right? Interfere with viruses. So they're known to interfere with viruses. And if you look at the types of interferon, there are 3 types of interferon: type 1 interferon, type 2 interferon, and type 3 interferon. And type 1 interferon is interferon alpha and beta, and there are 14 other subtypes, which you don't have to worry here. And there's a type 3 interferons, which is recently discovered interferon, It is also called interferon lambda. And what is intriguing about these interferons are they activate a JAK-STAT signal, and this JAK-STAT signal goes and binds to the genome or the DNA and induces interferon-stimulated genes called ISGs. There are multiple ISGs, there are hundreds of ISGs, and I've shown you a couple of ISGs like MX1, OS1, ISG15. These are few ISGs, but there are hundreds of ISGs which are important for to control viral infections. So what these ISGs do when they see interferon is they go and block the virus. So my lab is interested in when a cell sees a virus, how is interferon made, and how does interferon signal through a cell which is infected, and which ISGs are important to block the virus, and how do they function to block it. So I'll give you a couple of examples. Because it's just a 1-hour webinar, I just want to give you a flavor of what we do in the lab, as well as give you an outline of how these work, right? So whenever you get a viral fever, usually you feel sick, right? You feel you have a fever, you feel chills, you have a headache, you feel crappy. And that is not because of the virus. It is because of the interferons which make you sick, right? And that's good news. If you're feeling crappy, it's because your interferons are actually working, right? So interferons induce these hundreds of ISGs, which are the foot soldiers fighting these viruses. Okay, so that is the primer for interferons, and I'll give you one example of one of the most serious diseases called hepatitis C virus. So as you all know, it's a common bloodborne disease. And at least in US, there are 3.2 million people who get infected. And usually this is through needle use, drug use, but also it could be transmitted by blood transfusions, which happened a long time ago, which now doesn't happen. But there are 170 million people worldwide with this infection. So at least in US, before the new antiviral drugs, you had 8,000 to 10,000 people dying of untreated hepatitis C virus. And these drugs, the recent drugs are good, but they are $120,000 per person per treatment, which is an exorbitant amount of money, and also Americans will not be able to pay it. So these drugs are effective, but it's really expensive. What happens when you go untreated? You have usually it infects the liver, and you end up untreated. It changes to cirrhosis and to hepatocellular carcinoma. And eventually, if you don't get a liver transplant, the patient dies. So one thing was very intriguing in Hep C was certain people would clear the virus very easily without any treatment, and others needed extensive interferon therapy for clearing the virus. That is one situation. The other situation was the people who got interferon treatment, certain people would still be resistant and still have the virus, and others would clear the virus. And this was intriguing because interferon treatment also cost around $30,000 to $40,000 a year. And as you know, as I told you, giving interferons, you feel sick. And imagine feeling sick for 48 weeks and still you don't clear the virus. So this was intriguing, and two groups really spearheaded to ask this question: what is the difference between these two groups of people who either clear the virus or who don't clear the virus? So they went and sequenced these single nucleotide changes, which I was mentioning, across the genomes of 2 to 3,000 patients and healthy controls and the people who cleared the virus and the people who did not clear the virus. And what was very intriguing, and I'll show you this in the Manhattan plot, and it's called the Manhattan plot because usually it looks like the skyline of New York, the Manhattan skyline. Usually you have multiple peaks, and what these peaks are suggesting is these are chromosome arranged across, and you can see the chromosome number. And what was intriguing is when they did this genome-wide association study, where they looked for SNPs in all these patients and patients who cleared and who didn't clear the virus, they found this one single nucleotide. And you can see the p-value, it's really high. And this one nucleotide determined a person clearing the virus and person not clearing the virus, right? A person who was infected cleared the virus, had a C, and a person who got sick and did not clear the virus was T. And now you can imagine this is like a wealth of information trying to figure out what is this one single nucleotide doing. So if you go and genotype people, at least in America, trying to look for this one particular SNP, so C versus T, what they found is the C frequency was very low in African Americans, but very high in European Americans or East Asian Americans. And if you correlate that to the clearance of the virus, African Americans were poor clearers of the virus, but East Asian Americans were really good at clearing the virus. Now you can see how this became a tool to figure out who should get a treatment and who should wait for a better treatment or who should find an alternative way of treatment, right? So this was really important discovery to the Hep C field. But the question was, what is this one nucleotide doing? So a number of labs worked for a number of years to figure this out, and this is where I was starting my lab. And we were also interested in understanding this because, interestingly, this SNP was in the interferon locus, and the interferon lambda locus. This is the type 3 interferons, which I was talking about. And this became interesting for us because of the inherent interest in interferon lambda and also hepatitis C infection. So long story short, there, as I can show you all the papers which were published on this thing, what we found, and these are, if you're interested, you can go and read the papers, which are already published. We found a single nucleotide polymorphism of that one nucleotide change. What happened is, if you had a T, that reduced the gene expression of interferon lambda, an important interferon. And when you had a G, you increase the amount of interferon, and the increased amount of interferon cleared the virus. It is simple as that. It is the gene dosage of interferon lambda. But that one nucleotide change made an impact on how we clear that virus. Okay, so that is one story of how one nucleotide can have an enormous effect. Now I'm going into why we need two antiviral interferon systems. I told you both kind of converge in the same region, but still, we still have two interferon systems, and you can see this from fish to humans, right? So we know that type 1 interferons are critical for viral infections. Our study and other studies in the field showed that type 3 interferons or interferon lambda are important for other viral infections like hepatitis C virus, norovirus, and recently influenza or flu. So we asked whether is there a redundancy or do they have specialized functions, and are there any biological differences between these two? So one thing we know is type 3 interferons receptors are only expressed in epithelial cells. It's expressed in liver, skin, gut, lung epithelium. But type 1 interferon receptor is expressed all over our body. Okay, so if you give type 1 interferon to a person, every cell in your body reacts to it, but type 3 doesn't. So that was one big difference, which was known before we started this work. So this work was done by Adriana Ferrero when she was a postdoc in my lab. 2 months ago, she just went and started her own lab at Ohio State University, and this was one of her major questions for this work. Okay, so the basic question was, are there any collaborative functions of type 1 and type 3 interferons? A simple experiment I'll show you from the paper which he recently published, and you can go and look into this paper. I'll just show you the highlights of the paper. And what we see here is we treated cells with either type 1 or interferon beta or type 3 or interferon lambda 72, 48, 24, 12, and 6 hours prior to infecting that cell with a virus called VSV. VSV is just a tool for us to work with. They're interferon sensitive. It can be manipulated, and it can help us understand the biology of viruses as well as the interferons. So when we did that, we found something really interesting. So if you give in type 1 interferon, you see early on, you see that there is protection of the cells. Usually, if the virus infects the cells, you have at least the VSV, you see lysis of cells, right? But you don't see lysis of cells early on. As the time passes, right, interferons have no activity. All these cells start dying, right? But if you give interferon lambda, it's the opposite. You see that early on, you don't see activity. As the time goes on, you see lambda is more active. That means to say, if you have both interferons, you're way more protected than having one or the other, showing a collaborative nature of these two types of interferons. That's why in evolution, I think these two interferons are still present. Okay, so I'll show you another snippet of data from the same paper by Emily Heeman, who said, okay, if these two interferons are so important, what happens if you give interferon directly into the lung? And I told you, when you give type 1 interferon to a patient, you feel sick, right? You have chills. It's because of the interferons. And Emily, who also has a lab, helped us with this experiment. What she did is gave either interferon beta, a lower amount of interferon beta, and a higher amount of interferon lambda into the lungs of mice, and then looked for the type of immune cells in the lung. And remember this, SARS-CoV-2 also infects the lungs, right? So what was intriguing is, if you give them just interferon, there is no virus here, you see a lot of immune cells coming into the lung, meaning a lot of inflammation-producing cells coming into the lung. But if you give them lambda, interferon lambda, there's not much compared to PBS. And if you see these mice in the cages, when they get in type 1 interferon, they are sitting in the corner, not looking happy, right? But interferon lambda received mice are happily moving around the cages. That shows that interferon lambda is not as toxic or producing inflammation. But both interferons are required, but looks like interferon lambda is more mellow, right? So Adriana did a bunch of molecular work to figure out why does type 1 interferons bring in immune cell recruitment versus why type 3 interferons don't. But they have their own benefits. Type 3 interferons can have a long-lasting viral resistance, but type 1 interferon is early, but also bring these immune infiltrates, which helps fighting infection, and also prime the adaptive immune system like B and T cells to make antibodies, and also have other memory type of immune cells, right? So both are important, but it looks like type 1 interferon causes inflammation. So this was an important paper, a seminal paper for the field. So the journal Immunity wanted to put this on the cover. So we were fortunate that a artist from Bombay, Falguni Gokhale, kind of rendered this idea of the collaborative nature of type 1 interferon in red and type 3 interferon in blue, and how they are coming together, helping clear the virus. And as Falguni tells me, she sees it as Sagar Manthan, where something good happens at the end, right? So these are the things what we know how interferons work. And coming to SARS-CoV-2, the field was really excited because we know it is infecting lung, and SARS-CoV-2 maybe interferons might help, and there are clinical trials going on. Some are using type 1 interferons, and some are using type 3 interferons, and the jury is still out. We are hoping to find out what these results are. But the recent 2 studies which have come out, one from Major et al. And one from Broggie et al., which were back-to-back in Science, what they found is interferons are actually bad. Okay, so there are some caveats to this. Because without interferons, you cannot clear the infection. That is still true. But because there is so much virus and there is so much replication, how much ever interferon is made is not helping. So you need interferons way earlier to clear the infection, but having interferons later on is not helping. So that is a very intriguing prospect. And Matt Hendricks in my lab wrote a preview at Immunity in the talking about these 2 papers and how this could be important to really think about how we use interferons using interventions in clinical trials. So there are 2 clinical trials going on. So we will wait and watch and see how interferons either help or hurt, depending on the time. And I strongly believe earlier interferons are actually going to help, and we should wait for the clinical data to see if we are right or not. Okay, so I told you about interferons, but I also told you interferons make hundreds of interferon-stimulated genes. Okay, so the question now in the field is, what are these interferon-stimulated genes doing? How are they actually affecting the virus? How are they actually affecting the viral replication? So I'll tell you one story by Johannes Schwerk, or he goes by Hannes. He's an acting instructor in my lab, and he studied one of these interferon-induced genes, which is known as a viral restriction factor, which is a known viral restriction factor called zinc finger antiviral protein. And in short, we call it ZAP. Okay, so I'll tell you a little bit about when the virus gets into a cell. And these it depends on the type of virus. Okay, I'll show you some common viruses, and at the end I will touch upon how SARS-CoV-2 could also be using these mechanisms. Okay, so when viruses get into the cell, the first thing they do is try to block the immune response. Okay, and what we find is they try to hide from the immune response within a cell, and they can do it in a couple of ways, right? So they can, at least positive sense RNA viruses, they have this very interesting way to borrow membrane, intracellular membranes, and hide their PAMs and PAMs within these membranes so that our innate immune responses cannot see them, right? Flaviviruses like West Nile virus, Zika virus, and hepatitis C virus, they borrow membranes from ER and Golgi and form this nice looking homes for themselves and replicate inside. Okay, and I'll give you an example today of alpha virus, which does it, is chikungunya. As you guys all know, chikungunya is a problem in India. And Sinbis, what they do is see the lysosome, and they hide in the lysosomal membrane. These small drops are the lysosomal membranes. And if you think about other viruses like picornaviruses like EMCV, Coxsackie virus, or polio, they utilize the Golgi membrane for their replication and also to hide. Okay, so we asked, can immune genes or ISGs or interferon stimulated genes get into these regions? And will they be able to access these regions and neutralize this virus? Okay, so before getting into it, I just want to give you a primer on alternative splicing, and I know you guys all know about it. Alternative splicing is where the exons kind of shuffle in different way, right? Sometimes you lose one exon or the other, and you can form different splice forms. And these splice forms make a little bit different protein, and this different protein could either have modulation of dosage of activity, gain of function, or loss of function. And there's a lot of data on this in the literature, which you could go in, and we have published a few about this. But it is intriguing that because the virus replicates so fast compared to humans have to devise strategies to counter the pathogens, and we think alternative splicing is an easy way to counter the pathogens quickly. Okay, so I'll tell you an example of that. Okay, so I said zinc finger antiviral protein, it's called ZAP, and ZAP is intriguing. It's a protein which has zinc fingers at the N-terminal, and these zinc fingers are helpful to bind to the RNA. Okay, and also it has certain other domains, and I'll come back to it, but what you need to see is the ZAP has two isoforms, ZAP short and ZAP long, and ZAP long has a PARP domain, okay? And this PARP domain has important activities, but in this case, the PARP domain is inactive. So what did we know when we came into this work? There are a number of labs for a number of years have worked on this and figured out that this antiviral protein is really important in protecting us against HIV and alpha viruses. Like I said, chikungunya and Sinbis, okay? So the way it does it, ZAP goes and binds to the viral RNA, brings in degradation complexes, and degrades the RNA of the virus. So if you don't have the RNA of the virus is not made. So very simple strategy. Goes, binds to the RNA, viral RNA, and degrades it, right? So when we got into this for a very different reason is we wanted to know, because I told you that we are looking at interferons, and we were looking at the interferon UTR, which had a number of AU-rich regions, which is irrelevant for the story. But when we're looking for it, we did some mass spec to figure out what RNA binding proteins bind to this host UTR, the host interferon. And we did bioaccumulation, pull down, and mass spec. What we found is there were multiple proteins binding to interferon lambda UTR. And the first protein which came up, which was highly significant, was a protein called ZAP short, which is zinc finger antiviral protein, the shorter isoform, which does not have the PARP domain. So this was intriguing because I told you that this protein was important for its antiviral activity. And we were pulling this on a host mRNA, right? An interferon mRNA. So we tried to first figure out how this protein is alternatively spliced. And I'm not going to bore you on the details, but what I will say is it has its own polyrelational signals, and that makes it easier for it to alternatively splice. But what is important in this is when it gets alternatively spliced, the short isoform loses the terminal 4 exons, so it is shorter and does not have the PARP domain. Okay, that is the most important thing you need to get out of this. Okay, what is more intriguing is these both form mRNAs and also protein. And when we looked at the expression kinetics of this, we found that ZAP short is interferon inducible. Okay, like I said, it is an ISG, it is interferon inducible. But ZAP long was not. ZAP long is always expressed. So I'll show that in this Western blot. As you can see, these are two cells, one with no modification, one where it has deleted its IFNAR receptor so that it does not sense interferon. What we found is ZAP long is expressed all over, but ZAP short is induced later in the time point from 24 to 48 hours. Okay, so ZAP short looks like it's an interferon inducible protein, but ZAP long is not. So we started asking the question, what does ZAP short do and what does ZAP long do? And we use some immunoprecipitation biochemical methods to transfect these either ZAP short or ZAP long in a cell and induced interferons in these cells and did a IP of the proteins and then looked for the interferon messages. And what we found was very intriguing. ZAP long, even though it had the same zinc finger binding domains, because it is alternative spliced, would not bring down the interferon mRNA. But ZAP short, on the other hand, bound to interferon mRNA very tightly. Okay? So the next question was, if it is binding to host RNA, what is it doing? So we knocked out this ZAP long and ZAP short.